myocardial injury is something that patients in the operating room may experience, and that significantly contributes to the morbidity and mortality of our patients. So I've had a long-standing interest in studying heart attacks, and something that we've always been curious about was this observation that patients who would experience a heart attack in the early morning hours would have a much more severe myocardial injury than patients in the afternoon. This has been established in the 1980s through research that came out of, I think, Harvard Medical School, publications in the New England Journal that found that early morning onset myocardial infarctions had more often fatal arrhythmias, were more often associated with heart failure and overall bigger sized heart attacks as compared to heart attacks that occurred in the afternoon. That led to that understanding that there is a circadian or daytime dependent variation of the severity of myocardial injury, but it was not well understood what are the molecular regulators and what would determine these differences. And we thought if we could identify sort of the endogenous protective pathways that are active in the afternoon but seem to be absent in the morning, that could give us some new ideas how to prevent or how to treat myocardial infarctions, sort of putting people actively in this afternoon window rather than into this morning window. We didn't just randomly select some proteins, but instead we did screening studies, unbiased approaches that would lead us to these molecules. And we kind of started with screening studies in patients that had cardiac surgery and some degree of cardiac injury in the morning versus the afternoon. We were very fortunate that through a collaborator, through Danny Mühlschlegel, who was at that time was at the Brigham and Women's Hospital at Harvard Medical School, did a study where they performed biopsies of the left ventricle in patients that are having open heart surgery. And we looked for patients who had that done in the morning versus patients who had that done in the afternoon. And we did an unbiased screening approach and wanted to see, are there any differences in the expression of molecules that could be critical in this? So we kind of set up a human model of circadian rhythmicity of myocardial injury. And those studies led us to identify BMAL1 as the key differently regulated molecule. And we also set up a mouse model where we would do heart attacks at different times of the day. And similar to those studies in human patients, we found also in our mouse model that BMAL1 was the top hit. So that's how we ended up with BMAL. The question how we ended up with HIF2 alpha was sort of a second step. So BMAL is a transcription factor, and it doesn't function alone. It needs a partner to tango with, so to speak. And usually that partner for BMAL1 to tango with is a gene that is called clock. But in mechanistic studies, we found that actually it's not clock that is doing this together with BMAL, but a hypoxia-inducible factor, HIF2 alpha. And if you look back at it, it's actually not really surprising. So if you think about what are the typical aspects of a myocardial infarction and daytime variations, there's one element where there are differences in the daytime. So it's not surprising that BMAL1, which is a key, actually the key transcriptional regulator of the circadian rhythm, is functionally active. But the other thing that happens during myocardial infarction, if you think about it, what happens mechanistically is that the blood flow to an area of the heart is interrupted. And that area of the heart suddenly becomes extremely hypoxic. There's no more oxygen coming from the coronary artery into this area. And so these tissues become profoundly hypoxic. And the key regulators of adaptation to hypoxia are transcription factors that are called hypoxia-inducible factors, HIFs. And so it turns out that BMAL1 forms an active transcriptionally active heterodimer with HIF2 alpha. So we have, on the one hand, a key regulator in circadian rhythm, and on the other hand, a key regulator in hypoxia adaptation, forming a transcriptionally active complex that then drives cardioprotective responses that are absent in the morning, but highly active in the afternoon. I think if you want to answer a question like this, you usually try to look at it from a lot of different perspectives to sort of get a complete picture, and also see if the answer that you're giving fits into these different perspectives. So one perspective we took as a model system was human tissues of patients who had cardiac biopsies in the morning and the afternoon. I think it's important to have this human element because we don't want to be doing research that only applies to mice or to in vitro systems. As a second step, we had a heart attack model of circadian variation of infarcts. This is on many levels, a really painful, so to speak, or challenging model. Number one, in order to do a heart attack in a mouse and then to let the mouse survive and be able to observe it for up to 2 weeks, you have to do sort of open heart surgery on a mouse with a heart that beats at a rate of 400, and you have to put a suture around the left coronary artery, tie it off, and then allow reperfusion later on. And that is technically extremely challenging. It takes most people 6 to 12 months to learn this model. And the first author from our research paper, Wei Ruan, who's actually a cardiac anesthesiologist, was able to perfect this model. But then on top of that difficulty, you have to do it in a way that if you want to address circadian rhythmicity, that you do infarct sizes at different daytime periods. So she did heart attacks at 3 a.m. in the morning, and she did them at 3 p.m. in the afternoon. So she, together with her team, they came here and spent uncounted nights in the laboratory inducing myocardial ischemia and reperfusion injury in mice at 3 a.m. in the morning and in controls then in the afternoon. So this was our our mouse in vivo model, where we could actually see that similar to the human, mice that had infarcts in the early morning had much bigger heart attacks. And also when we followed up on them with echocardiography, their heart function was much more affected than mice that were operated on or had infarcts in the afternoon. Then as a third model, we used in vitro studies of ambient hypoxia. So if you do research, you have human components, you have a murine model, but you also need to be able to control your conditions very well. And that is best done using ambient hypoxia as an in vitro model. So we put cardiac myocytes into a hypoxia chamber where we could reduce the oxygen percentage to 1%. So these cardiac myocytes became profoundly hypoxic. And then we used those cells to study the interaction between BMAL1 and HIF2 alpha. But then I think on another level, a very important aspect of our work was that we used a system that would allow us, using cryo-electron microscopy, to really visualize the interaction between BMAL1 and HIF2 alpha. Dr. Quang-Lei Zhao, who's a biochemist at UTHealth, is very experienced with structural molecular biology, and he collaborated with us and said, I'm going to go ahead and figure out how these two molecules actually fit together. How do they look? And something very surprising that he identified is that usually BMAL partners with clock, but in our case, it partners with HIF2 alpha. And BMAL had to undergo a structural rearrangement, which was unexpected, to allow this interaction between BMAL1 and HIF2 alpha to form an active heterodimer, a complex of those two molecules, one from the hypoxia side, one from the circadian rhythm side, to then induce target genes such as amphiregulin to provide cardioprotection from ischemia and reperfusion injury. Having shown that there is an interaction between BMAL1 and HIF2 alpha on a cryo-EM level. And we also did functional studies where we knocked out one or HIF2 alpha in genetic models. And we found that this difference in heart attacks, the fact that the afternoon heart attacks are smaller and outcomes are better, that was gone after genetically deleting BMAL1 or HIF2 alpha. But obviously, the question then becomes, how does that work? What do those transcription factors do to provide cardioprotection, particularly in the afternoon, as opposed to the early morning hours? And so transcription factors usually function by inducing target genes. And so we looked for different target genes. We had previously done studies with HIF2 alpha and had done microarrays, and we found that the top hit on those arrays was a gene called amphiregulin, or as you said in short, ARIC. Amphiregulin is a growth factor that activates a receptor, ERK-B1, that sort of promotes the survival of myocytes. It prevents that they are going into a death cycle following myocardial injury. It prevents myocyte apoptosis. So we looked carefully at that question if potentially amphiregulin might be the answer, might be this transcriptional target that HIF2 alpha and the BMAL1 complex could be regulating. And we found, number one, that amphiregulin release in the myocardium was transcriptionally regulated in a circadian fashion with higher levels at certain time of the day versus others. So it followed that idea that it could be regulated by this complex. Moreover, we looked at what happens if I delete or genetically knock out either HIF2 alpha or BMAL1. And in both instances, the circadian variation of amphiregulin was abolished. And then finally, we did functional studies where we took mice that had a genetic deletion of amphiregulin and looked at their heart attacks. And we found that similar to mice with myocyte-specific HIF2 alpha or BMAL1 deletion, this variation that we had seen with large infarcts in the morning, small infarcts in the evening was gone after amphiregulin deletion. In fact, the amphiregulin knockout mice had similarly big infarct sizes in the afternoon. And eventually we also went at this from a therapeutic perspective. We gave recombinant amphiregulin, which was known to be cardioprotective, but the cardioprotective effects was particularly dominant in the afternoon during a time when sort of those big heart attacks occurred. So that's how we identified amphiregulin as a target. And we think that the mechanism of how these daytime variations can be explained is that HIF2 alpha and BMAL1, one hand from circadian rhythm, BMAL1, one hand from hypoxia signaling, HIF2 alpha, form this complex that then regulates amphiregulin expression. And amphiregulin, by activating an amphiregulin receptor, prevents myocyte death during myocardial infarction or myocardial injury. I think, on the one hand, this is an interesting molecular explanation of why heart attacks are bigger in the morning than in the afternoon. To be honest, probably there are also other mechanisms involved in this. We're not claiming that this is the only mechanism, but it seems based on the results that we have from our studies that this is a key mechanism that explains these differences. But as a physician scientist and practicing anesthesiologist, obviously we want to do things that can be used to help our patients. And so by studying this pathway, we observed several things, and there are several ideas that could potentially be used for cardioprotection. We did some studies using a circadian rhythm enhancer, nobilitine, that would drive and enhance circadian rhythmicity. It would drive BMAL1, and we saw a benefit with that. A second approach that we took was using amphiregulin, recombinant amphiregulin that we gave. But probably for me personally, the most promising idea that comes out of those studies is using a pharmacologic HIF activator. So hypoxia-inducible factors were identified in the early 1990s and were rewarded with a Nobel Prize in 2019. Three famous physician scientists were awarded this Nobel Prize for physiology and medicine. And in that context, it is important to realize that over that time period, there was a lot of effort from scientists and also from pharmaceutical companies done to develop pharmacological agents that would alter this pathway. And we now have pharmacologic HIF activators that are FDA approved for patient treatment. These HIF activators are approved for treatment of renal anemia. HIF is a main driver of erythropoiesis. So if you have low blood levels, erythropoietin is stimulated through a HIF-dependent pathway, and you can use a HIF activator to kind of boost this pathway, particularly in patients who don't have good kidneys. They have lower erythropoietin levels, and so these FDA-approved drugs can help there. And something that I want to consider for our patients in the perioperative setting is, can we use these type of HIF activators, such as vadadustat is one of those drugs, can we use them in clinical trials to see if we can attenuate or prevent myocardial injury, which is, as I had talked about early, one of the big and leading causes of perioperative mortality. Dying within 30 days of an operation is considered the third leading cause of death in the United States and in a lot of other Western countries. And one of the main contributors to myocardial injury is perioperative myocardial injury. It very often is undetected, but if we would have a way to prevent this from happening by making myocytes more resistant to injury and to hypoxia or ischemia, that would be phenomenal. And as similar as to what we had done in this paper about BMAL1 and HIF2 alpha, we would like to consider a HIF activator such as vadadustat in a preventive fashion to give to patients that are having heart surgery or having major surgery or are at high risk for developing perioperative myocardial injury, to prevent this from happening and thereby improving outcomes in patients that are having surgery.